This video will introduce gene regulation in eukaryotes and dive into one of the three main mechanisms of this process. So let's begin with a basic question. Why regulate gene expression in our cells? While the answer to this question is actually quite complex, in the most basic sense, we need this process for a few main reasons. The first is that it allows us to respond dynamically to changes in our environment. Another reason is due to our complex development and multicellularity. With a few exceptions, all our cells contain our entire genomes, but we don't need the gene products from our entire genome in each of our cells or at each stage of our development. Gene regulation allows for things like cell differentiation and complex changes during life stages. It's therefore a crucial part of being the complex multicellular beings that we are. The regulation of which proteins are produced in a cell can happen at multiple levels within the central dogma. They can happen before transcription even begins, all the way to editing the final protein after translation. Here, we will focus on regulation at the beginning of transcription. We will begin with regulatory transcription factors. Regulatory transcription factors are proteins that influence the rate of transcription, and these are divided into two categories based on their mode and location of action. General transcription factors act directly on RNA polymerase at the core promoter and affect whether it can get set up and progress to the elongation stage of transcription. These include some of the common transcription factors we discussed previously in our video on transcription. Regulatory transcription factors typically bind upstream from the promoter and regulate the ability of RNA polymerase to initially bind in the beginning of transcription. Regulatory transcription factors recognize cis-regulatory elements near the promoter. When these regulatory transcription factors bind, they can affect transcription of a gene by either enhancing or or increasing the rate of transcription, or silencing or decreasing the rate of transcription. While our setup so far has been relatively straightforward, the reality is that it is often way more complicated with multiple activator and repressor proteins, along with a host of other factors that modulate this process. This multifactorial control is referred to as combinatorial control. It actually gets even more complex than these regulatory transcription factors because they can work in concert with changes in nucleosomes and DNA methylation to regulate gene expression. We will discuss these other two modes for gene regulation in upcoming videos. Our regulatory transcription factors generally take the form of either enhancers or silencers. These are quite important for the efficiency of the whole transcription process. And for example, enhancers can increase the rate of transcription by 10 to 1,000 fold. While we've established how complex this process can be, let's zoom back in on the regulatory transcription factors for a moment and look at two common modes of action for these elements. The first is regulation of the general transcription factor directly or through co-activators. In this mode, the binding of our core transcription factor complex will be either enhanced, as seen here, or repressed, as seen here. In this figure, the repressor protein is blocking the binding of a transcription factor to the promoter. A second type of is regulation via the mediator. If you recall from the transcription video, the mediator is a transcription factor that interacts with upstream elements to affect the rate of transcription. Some of these regulatory transcription factors thus interact with the mediator to activate transcription, as shown here, or repress transcription, as shown here. In the figure here, the repressor protein is blocking the mediator and transcription factors from proceeding on to the elongation step. While regulatory transcription factors work to affect the rate of transcription, they themselves are affected by a variety of factors. Commonly, they are modulated in three ways. The first, shown here, is the binding of small effector molecules such as a hormone. In this example, the transcription factor can only bind in the presence of the hormone due to how it changes the transcription factor structure. Thus, the presence of the hormone regulates the rate of transcription indirectly by regulating the transcription factor. A second way is through protein-protein interactions. In this example, both protein subunits are required for proper binding of the transcription factor, which will affect transcription. The last way is through covalent modification, such as phosphorylation, which can affect how the transcription factors interact with the enzymes at the core promoter. In the next video, we will continue to explore ways in which gene expression is regulated.